Welcome to The Explainer. Today, we are diving into the incredible story of the tools that let us edit the very code of life itself. And believe it or not, this journey doesn't start in some high-tech, futuristic lab. It starts with bacteria fighting a war against viruses. You know, sometimes the most complex ideas are explained best by a child. And I don't think there's a better starting point for our story than this quote. It's from Sylvia Arbor when she was just 10 years old, describing her father's Nobel Prize-winning work. She absolutely nailed it.
So let's rewind to the 1950s. Scientists were looking at this puzzle. They noticed that certain viruses, we call them bacteriophages, could wipe out one type of bacteria, no problem. But when that same virus tried to infect a different strain, it got stopped dead in its tracks. The bacteria were somehow fighting back. But how?
Well, the mystery was finally cracked by Werner Arbor, Hamilton Smith, and Daniel Nathans. And it turns out that little girl's description was spot on. They found the servants with scissors. We call them restriction enzymes. And this discovery wasn't just a small step. It was so fundamental to everything that followed that it earned them a Nobel Prize. Now you have to remember, this wasn't a tool nature designed for us. This was a bacterial defense system perfected over billions of years. These little molecular scissors evolved to do one thing: find the DNA of an invading virus and shred it to pieces. And to make sure they didn't accidentally chop up their own DNA, they put these little chemical "do not cut" signs on it. Just a brilliant strategy that we were about to borrow. Okay, so finding nature's toolkit, that's one thing. But figuring out how we could actually use those tools for ourselves, well, that's what lit the fuse on a full-blown revolution, all happening inside a tiny test tube. And the precision of these things is just mind-boggling. An enzyme will scan along a strand of DNA, reading the letters, and it will do nothing until it finds its one specific target sequence. It's like having a key that can find and unlock a single specific door in a city of millions. But here's where it gets really, really clever. The way they cut matters. Some enzymes make a nice clean cut straight through. We call those blunt ends. But others make a staggered cut, and this leaves these little single-stranded bits of DNA hanging off the end. They were given the perfect name: sticky ends. And those sticky ends were the whole ballgame. Why? Because that little overhang is desperate to pair up with a matching partner. So if you take DNA from, say, a human and DNA from a bacterium, and you cut them both with the same enzyme, their sticky ends will match up and stick together perfectly. You add a little molecular glue, another enzyme called DNA ligase, and boom! You've just stitched together DNA from two different species. This simple cut and paste ability launched the entire field of biotechnology. Seriously, this is the moment humanity went from being just readers of the genetic code to being writers. All of a sudden, we could take a human gene, pop it into bacteria, and turn them into tiny little factories, churning out life-saving medicine, like the human insulin that completely changed diabetes treatment back in 1979. But these first-generation scissors, as powerful as they were, had a pretty big flaw. They recognized really short sequences of DNA, maybe just 6 letters long. Now, in a tiny virus genome, that's fine. But what about in the human genome, with its 3 billion letters? Well, that 6-letter sequence might pop up millions of times. It was like having scissors that just cut everywhere. So this became the next great quest in genetics. It wasn't enough to just cut anymore. We needed a way to tell the scissors exactly where to go. We needed to be able to pick a single, precise address out of that massive genetic landscape and make just one cut right there. This led to some seriously clever but also seriously complicated tools. You had things with crazy names like zinc finger nucleases and TALENs. The concept was brilliant. You basically bolt a custom-made protein that grabs onto a specific DNA sequence onto your cutting enzyme, and they worked. But my gosh, they were a nightmare to build. It was so difficult and expensive that only a handful of specialized labs in the world could really do. So you can see this progression, right? We went from just observing bacteria in the 50s, to using their basic tools in the 70s, to building our own incredibly complex machines in the 2000s. Each step was a major leap forward, but the true revolution, the tool that would put this power into the hands of everyone, was just around the corner. And then in 2012, everything changed. Researchers, once again taking a cue from bacteria, figured out how to adapt a different immune system called CRISPR. And in what felt like an instant, gene editing became simple, cheap, and unbelievably powerful. The absolute genius of CRISPR-Cas9 is how simple it is. You've got two parts. First, you have the Cas9 protein. That's the scissors. But here's the key. You never have to change the scissors. It's a one-size-fits-all tool. All you have to do is give it a new set of instructions for each job.
And those instructions, they come in the form of a little molecule called a guide RNA. It's basically a genetic GPS coordinate. You program this guide with the address of the DNA you want to find, hand it to the Cas9 protein, and the protein just goes right there and makes the cut. It's that easy.
The whole process is just beautiful. Step 1, the guide RNA homes in on its target. Step 2, the Cas9 protein snips the DNA. And step 3? Well, step 3 is where the cell's own repair crews come in to fix the break. And that's our chance. We can trick the cell into either disabling the gene, or by giving it a correct template, we can have it fix a genetic typo for us. And this table really says it all. You went from a process that was incredibly difficult and time-consuming to one that's based on simple RNA pairing. Targeting a new gene is now as easy as designing a new guide RNA on a computer and ordering it online. This didn't just improve gene editing, it democratized it. So now that we have these powerful, programmable, and easy-to-use genetic scissors, the world has opened up. So what's actually possible? And what are the huge responsibilities that come with this power? The potential is honestly breathtaking. This isn't science fiction anymore. Right now, there are clinical trials using CRISPR to treat diseases like sickle cell. People are working on ways to fix the mutations that cause cystic fibrosis, to maybe even cure HIV, and to design crops that can resist disease and feed a growing planet. But, and this is a really big but, there are still huge hurdles. We have to be absolutely sure these scissors only cut where we tell them to. An accidental off-target snip could be a disaster. Then there's the delivery problem. How do you get the CRISPR machinery into the right cells inside a person? And, of course, there are the massive ethical questions. Just because we can edit our own DNA, does that mean we should? Where do we draw the line? So when you step back and look at this journey, it's just incredible. It started with simple curiosity about bacteria, and that curiosity led us, step by step, to a tool that lets us rewrite biology itself. The science is moving at a blinding pace, but that leaves us with the most important question of all, one that isn't just for scientists. We have the scissors. Now, all of us have to decide, what are we going to write?